Welcome to the Gladden Longevity Podcast with Dr. Jeffrey Gladden, MD, FACC, founder and CEO of Gladden Longevity. On this show, we want to answer 3 questions for you. How good can we be? How do we make 100 the new 30? And how do we live well beyond 120? We want to help you optimize your longevity, health, and human performance with impactful and actionable information. Now, here's today's episode of the Gladden Longevity Podcast.
Happy New Year, everyone. I'm recording this on January 2nd. I'm not sure when you'll hear it, but I have a guest with me today that I've known for 5 or 6 years who I think is really a pioneer in the space of functional medicine, but also really dovetails really nicely with longevity medicine. Her name is Sharon Houseman-Cohen, and she's an MD that trained at Harvard. She has a master's and an MD. She's basically been involved with genetics for a long time. And she and her co-founder started a company called IntellexDNA, which is a company that we've been using for the last 5 or 6 years at Gladden Longevity. It's one of the primary tests that we do for every client that we see because it has such good information, such good recommendations, and they continue to expand their testing and their recommendations. One of the things I love about the test is it's an evergreen report. So if they update information on the genes, you keep getting the updates, so to speak. It's really, really nice. Now, if they come out with brand new genes or something, I think there's another test that needs to be done. But in essence, it's an evergreen sort of report. I think you're going to love this conversation because you're going to hear just how foundational genetics are to understanding your future and how to optimize your health and longevity. It was really a lovely conversation. Welcome, everybody, to this edition of the Gladden Longevity Podcast. I'm your host, Dr. Jeffrey Gladden, and it's a new year, a new day, and lots of interesting thoughts around the new year, quite honestly. One of the things that I've really committed to this year is doing hard things. I've kind of been cultivating a love of doing hard things, and I'm having some really nice insights around that in cultivating doing hard things and a love of doing hard things, we tend to shy away from doing hard things. But what I found is that when you do hard things, you actually elevate to another level where it gets easier again. What I mean by that, it's a little bit like a jet airplane, right? So jets don't fly well at low altitude. They expend energy to get to high altitude, but at low altitude, 10,000 feet, 15,000 feet, they're very inefficient. They don't fly very quickly. And so it's hard to climb to 35,000 feet. But when you get to 35,000 feet, everything is efficient, everything is good. So I really embrace this concept of doing hard things and as a means to getting to a higher level of flow state, if you will. So that's kind of one of my things that I'm working on for this new year. Speaking of doing hard things, I have a guest with me today that I'm really excited to have on the show, Sharon Houseman. Sharon and I have known each other probably for maybe 5 years now or something. We just did a TV show together on a particular supplement that's quite interesting called Vinia. But talking about doing hard things, Sharon's really been decoding genetics for clinicians, if you will, for clinical practices. And she's done a fabulous job of this. I don't know of anybody else that's doing a better job of this. And so I think with genetics and being kind of the initial substrate that one needs to look at and take into account when you're looking to optimize your health, your longevity, your performance, it's really kind of a foundational piece. And so with that, as a brief intro, Sharon, thanks so much for joining us here.
It's my pleasure. Thanks for having me.
Yeah. So to start with, I've heard you make the distinction between genetics and genomics and SNPs and genes and things like that, just to kind of tee the audience up so everybody's on the same page. Do you want to talk a little bit about that?
Sure. I think that the difference between genetics and genomics is important to understand because your genomics is your book of you. So it doesn't mean anything is wrong. It's just how you make every protein in your body. But a genetic disease is different than a genomic disease. So a genetic disease is something like Huntington's disease, Tay-Sachs, cystic fibrosis, sickle cell, where if you have 1 or 2 copies, depending on if it's dominant or recessive, you get the disease. But chronic diseases, the things that affect our longevity, heart disease, hypertension, obesity, osteoporosis, even things that affect inflammation and our ability to remove toxicants, that's not genetic, it's genomic, meaning there's no one gene that causes heart disease. There's no one gene that causes diabetes or cognitive decline. These chronic diseases are the influences of these small changes in your DNA. So that's what genomics is. The genomics is the study of the small changes in your DNA, kind of like one letter in a book that can be changed and can then have an effect on your risk, but it's not causative. So you have to have a lot of things simultaneously happen to develop heart disease. And genomics also you can respond to much better than genetics. Genetics is what we often many times we'll call genetic diseases being pathologic. There's nothing you can really do about it. If you have the genes for Huntington's disease or sickle cell, short of CRISPR, you can't get rid of it. But if you have genes that make you at higher risk because of your genomics to heart disease or diabetes, that's what you and I do in our practices as physicians. We say, how can you address your genomics? Because everybody has things in their family so that you don't have to have the same things happen to you that happened to your family members.
Yeah, I think it's important for people to understand that I've never come across, and I suspect you've never come across, somebody that has quote unquote perfect genetics or genomics, right? Nobody out there has that. Everybody has a constellation of different SNPs and different predispositions, if you will, to gaining weight or having osteoporosis or creating anxiety or dementia or all these different disorders that we live with. And a lot of the tendency for that is really in part a function of the environment, if you will. If you orchestrate the environment in a proper way around understanding your genomics, if you will, then you orchestrate the environment around it, you can actually minimize the risk of developing these things, which I think is one of the exciting things. And I think one of the things that you've really done extremely well in your reporting with IntellexDNA is how to basically shape the environment to counteract, mitigate, minimize a risk that you may be carrying genetically. So pretty interesting.
Yeah, absolutely. And I think that that's really important for those of us in medicine. We're taught at a very early stage in our career, don't order a test unless there's something you can do about it. And so the problem I was trying to solve when I was developing IntellexDNA is the fact that there was not genomics that was made for physicians and clinicians to help us improve outcomes with chronic diseases, and that the genomics that was out there, for the most part, nobody had taken the time to do the research to kind of figure out how do you modulate these gene variants in a very specific way. So that's kind of been my area of research.
Yeah, that's right. So let's say somebody does an Ancestry or 23andMe or whatever, and they get their health reporting or they go through another, online kind of genetic test. Typically what you get back is you get back a set of potential proclivities that you have or tendencies that you have, but you don't really get any recommendations about how to mitigate those. The recommendations are very limited. And then even you can talk about the quality of gene testing, which I think is so important also, right? How many times you oversample the genome to make sure that the genes that you're reporting have the integrity and the fidelity to actually be accurate, right? Because it's possible to do genetic testing and get inaccurate results. So you might want to contrast IntellexDNA with some of the more popular things in terms of the quality of the studies that are being done and things like that.
Yeah, I think that there's a couple of points that you brought up. One is the ability to know that the data is accurate. And we do validation. And we are a different class under the FDA. We're a clinical decision support tool, so we are held to higher standards. And we do run, each gene variant is run an average of 200 times. And we throw the data out if we don't get at least 30 copies. And that's the standard for if you were using a test for someone's cancer DNA or anything that was kind of a prescription type of DNA testing. So accuracy is important. Now, it doesn't mean that direct-to-consumer tools don't have any accuracy. It means that if you download their raw data, they're not guaranteeing the accuracy of that downloaded raw data because they only validate on the ones they report on. That kind of brings us to the other kind of side of that coin. If you were to do a direct-to-consumer test, they're really designed for ancestry and other purposes, not really so much for health, so that you might get a report on cognition that you have ApoE4, or maybe you're ApoE3. But it's not enough information to really help you optimize your memory because the ApoE4 gene interacts with many, many other genes. And so many of these other combinations can make you have either more or less risk. So there are I have ApoE4 individuals in my practice that are in their 80s that are doing extremely well in the 90th percentile for cognition with very short protocols. And then I have ApoE3 individuals who have cognitive decline that we have to do quite a bit to get them doing well. Same thing with diabetes. The direct-to-consumer tests generally will report on TCF7L2, which is a gene that codes for something called incretin, which is a hormone made in our gut, essentially, that helps us release insulin. And that's important in diabetes. Having that can more than double your risk. But 40% of the population have one copy of that, and there's many other genes that add to the risk. And so when you have the ability to look at the top 10 or 15 genes that relate to a particular disease state, that's going to give you more ability to kind of hone in on it, what's going on, and when you have the ability to know how to modulate it. So TCF7L2, in a direct-to-consumer report, they can't give specific advice. So they'll say, oh, eat a lower glycemic index diet, and that's great. But if you knew that this herb called Gymnema sylvestre helps you make more incretin, which is what that codes for, or whey protein, you could change your diet with that. And then there's specific other things like American ginseng and other things that can, again, help directly with that gene. So I think that what we've tried to do at IntellexDNA is take genomics to the next level so that it can be used by physicians who are trying to help their patients and other clinicians that are licensed healthcare providers, but by clinicians trying to help their patients optimize health and being able to combine our medical knowledge and the knowledge of genomics to make it a more precision medicine approach.
No, I think that's right. We're so enamored with IntellexDNA that we literally do it on every client that we see. And we found it to be extremely helpful. And it's interesting because some people will come in and say, well, I've got ApoE4 and I'm really worried about it. And it's like, okay, well, where'd you get that information? Well, I got it from, Ancestry or wherever else. It's like, okay, well, I understand your concern. Tell me about your family history. Let's sit down. Let's do a more comprehensive test. Let's do the IntellexDNA. And then we break it down and we walk them through, the entire cognitive panel, if you will. And we look for ApoE4 and ApoC1 and TOM40 and things like this, things that we know that are going to really make ApoE4 an issue, right? And one of the things that's fascinating is you can have ApoE4 and actually have a normal risk for dementia, which is interesting if you don't co-inherit ApoC1, right? But what's interesting also is that you can have ApoE4, and if you know what to do about it, you can also revert your risk back to that of an ApoE3, right? If you understand a genetic landscape and then you understand plasmalogen levels and other things that are important here, you can actually bring people back. So it's been fascinating for us to work with people and help them do this. It's really kind of like a joy to actually look behind the curtain, see what really the issue is, see where their concern lies. It's like, well, it's not as bad as you think it is. Or actually, there's more here to talk about than what you thought there might be. Let's talk about this and how we're going to go about it. I'm sure you do this all day long in your practice also. And I think it's one of the things that as an audience participant today, you should understand that you really need to know this information. I don't think you can really go about managing and optimizing your health without it. And if you're suffering with something, whether it's high blood pressure, heart disease, or anything else, you really can't get there from the standpoint of healing or getting better or mitigating your risk for other events without knowing this information. I just see it as being sort of cornerstone type stuff. Do you want to talk a little bit about the story of IntellexDNA, how you and your partner founded this company, and kind of a little bit of your story too about how you got to this point?
Sure. So I was interested in genetics back in the 80s, which gives you an idea. I know you can't see me, but that you and I have both been in practice quite a while, Jeff. And
Yeah.
I was interested in genetics, worked in a genetics lab doing research in cancer genetics. I also did research in making drugs more specific for the receptors. And I was also interested in things around the world, like how things were being done in other countries, because I had been an exchange student to Japan. But I decided I was going to become a researcher. And I went to medical school to get a PhD in medicine, not an MD initially. That was back in the late 80s at Harvard. And I got there and I was there for about a year and a half. And I said, oh my gosh, I don't want to spend all my time on one topic my whole life. And that PhDs often spend a lot of time on one topic and sometimes even one protein. So I went to medical school and I kind of said, well, at some point in my life, I'll go back to research when I find the right thing. And what happened is, in the last decade, people started coming to me with their 23andMe, with their different reports and saying, can you use my genomics to help me prevent Alzheimer's? Can you help me prevent heart disease? I was doing a lot of brain science because that's what I was going to do my PhD in. So I have a lot of interest in that. And I was working with mold patients, and everyone would start to say, oh, can you use my 23andMe? I just got this done. And I started looking for tools to be able to use it as a physician and to use other genomics, and really could not find any tools that gave me the kind of accurate, actionable information that was specific about disease states, to be honest, about things that were clinically significant. And at the same time, I switched my practice to more of a precision private practice where I spent a lot more time with people. So I started doing it by hand, using people's genetics and trying to work out if they had a family history of heart disease or macular degeneration or whatever it was, cognitive decline, trying to use their genomics and do this by hand, and realized we couldn't do it by hand. That practice was Resilient Health. It was co-founded with my co-founder, Carol Billich. So Carol and I decided that we needed to start mapping out this genomics, and that it took us so much work. We were doing it initially for each patient that it took so many hours. It made sense to make it something available across the country. And so that was kind of how it started. And because with the brain, you have this plasticity and you have this ability to repair, you can see outcomes pretty quickly. So we saw our brain patients getting better. And that made us, go, we've got to keep doing this for other disease states. Obviously heart disease, it's more of a prevention. So you can't really tell that you're preventing heart disease other than the fact that nobody goes to the hospital. But you can tell when you're getting somebody's brain better. And so now we do it for a lot of brain science, everything from autism, not really what your listeners are worrying about because you're more at the other end of the spectrum of longevity, but depression, anxiety, cognitive decline, and then all the other things that affect the body, because of course your bones, your heart, your blood pressure, your inflammation levels, and it's just kind of grown and grown over the years as we take on new challenges, which you started to talk about, doing things that are hard. And I would definitely say this is one of the hardest things I've done, but one of the most rewarding.
Yeah, absolutely. That's very cool.
Oh, I did go back and get a medical degree somewhere in there. Sorry.
Yeah, exactly. Yeah. And you went from being hyper-focused to being kind of a family practice, right? Where you're kind of a generalist, if you will, in terms of, you went from a narrow focus to as broad a focus as you could. So that was interesting.
Yeah,
too.
I didn't want to be put in, after having been working in the PhD track, I really wanted the background of family medicine so that I could do whatever needed to be done, because I felt like America had made medicine so specific that someone was being said, go here, go there. But not everybody who complains of stomach pain really needs a gastroenterologist. There's a lot of things that can contribute to that. So I like to be able to untangle mysteries, and that really comes to the primary care doctor. So that's why I chose to become primary care.
So let's talk a little bit about some specific disorders that I'm sure the audience is interested in. So we've talked a little bit about cognitive decline, and I know that you just did a research project, I think with Dale Bredesen, if I'm not mistaken, and you're writing a chapter in a book also about some of this, I think. I don't know, I'm hypothesizing a bit here, but I believe that's the case. Do you want to talk a little bit about, many people, we've had Dale Bredesen on the podcast actually and had a conversation with him a couple of years ago, the end of Alzheimer's and kind of his leaky roof and basically fixing all the different pieces, which quite honestly, we agree with. We don't think there's one solution to any problem, quite honestly. It really has to be multifactorial. But nonetheless, you were able to add a genetic piece to kind of what he was doing. Do you want to talk through a little bit about that and give people a sense of which genes you're looking at, how you're working with them, how they're interfaced with the protocol?
Sure. So yes, we are the genomics being used in Dr. Bredesen's 2 trials, the one that was published in the Journal of Alzheimer's last year and the ongoing trial now. And there is also a textbook chapter coming out in an Elsevier precision medicine textbook that I wrote, one chapter on the cognitive decline, and Dr. Bredesen wrote the other one. But I think that there's different categories. And for your listeners that have been listening to you for months and even years, a lot of these things are going to sound familiar. So there's genes that relate to brain inflammation, some of the different interleukins. Interleukin 1 can act very locally, and it can cause not only problems with increased brain inflammation, but people who tend to get more disc herniation. They get more inflammation and they might get less response to having prolotherapy. So every gene can have, more than one effect. There are genes that relate to mitochondrial dysfunction. You actually mentioned TOM40, which is mitochondrial membrane. And when you're looking at someone who has more of these mitochondrial things, even that has different levels, because there are genes that relate to mitochondrial DNA synthesis that we treat with folinic acid and B vitamins, and ones that relate to the mitochondrial membrane, where you're going to give more of the membrane lipids. There's, of course, things like MTHFR, but one of the things that we do differently is it would be great if genes were as simple as, add up this one and this one and this one, but there's a lot of interactions in the literature. So having MTHFR variant alone, 40% of the people have 1 copy and about 14% have 2. It does increase your risk of cognitive decline a little bit, about 14%. But if you combine it with having high levels of IL-6, which again, you can't just check your blood because of the blood-brain barrier, so you have to know the genetics of which gene turns on, which is in the promoter of that IL-6 and makes you have high brain IL-6. That combination of the 2 of them, the MTHFR and the high IL-6, can give you 2.5 times the risk of Alzheimer's or 3.7 times the risk of vascular dementia, which of course is probably even more common than Alzheimer's when you really start to dissect things. And that's a 270% increase. So you wouldn't be treating it in the same way that you would treat somebody where their issue is that they can't make choline. So that kind of brings to a different topic. There's a lot of different nutrients that relate to choline and zinc, and there's genes that control those that affect cognition. So those are some of the general categories. There's, people think of ApoE4 as the biggest risk for cognitive decline, but that's just because ApoE4 is so common. It's in 20-some% of the population. There are other genes that relate to microglial inflammation that have just as high of a risk, but they're much more rare. So I think that just gives kind of a sampling of there's so many things going on. And the exciting thing is when you know what's going on, you can again decide how do you want to proceed. And so I really am so thankful for Dr. Bredesen's work. But if you read The End of Alzheimer's, you leave that going, oh my gosh, I need to take 30 different things. And that can be a lot for some people. And it may not also be, cost effective. It may not agree with their stomach. So if you can instead go, okay, I'm a person that really needs to lower my inflammation, and I'm going to use sulforaphane for that. And I'm somebody who really needs to work on my microglia. So I'm going to use, EGCG from green tea or low dose naltrexone for that. And then I'm somebody who doesn't make choline well, so I'm going to take citicoline. You can kind of get where you're going with a lot less energy. And then we also look at hormone receptors because those are super important for cognition, hormones and hormone receptors, and growth factors like BDNF. And I just learned something really fun about BDNF today. I was working on a talk. BDNF is like fertilizer for your brain. It stands for brain-derived neurotrophic factor, and it helps nerves to grow. And if you have 2 copies of this gene that makes it so you can't convert the what's called pro-BDNF, kind of like the precursor, it's kind of like BDNF with an extra molecule on it, into the BDNF, it's not good. It gives you almost 3 times the risk of Alzheimer's. So almost the same as ApoE4. And exercise converts that pro-BDNF into BDNF. But they just learned why the pro-BDNF is so bad for you. Because Dale always said, that if you don't have enough BDNF, it can be synaptoclastic. So meaning it can take away synapses. Well, the reason not being able to convert the precursor, the pro-BDNF into BDNF, causes you to lose brain neurons is it turns out it stimulates the master switch called NF-kappa beta, but the master switch for inflammation. So if you let that pro-BDNF hang around, you will start to have more inflammation. And so I have a patient, and she's done really well. She's a professor, and she noticed when she was in her early 50s that she was starting to have some word finding difficulties and would use workarounds in her sentences. And she's an ApoE3/3, which is great, but she had that 4% of the population that had 2 copies of that inability to convert the pro-BDNF into the BDNF. And exercise helps to convert it. And really with her, exercise has been key, but she also had high levels genetically to predisposition towards interleukin-1 alpha. So with that high inflammatory switch, things like curcumin, sulforaphane, all of that were really, really key for her. And so everybody is unique, but I thought that was really cool to now they understand why you can't just sit on your bottom if you have BDNF problems, because you're going to actually create inflammation by doing nothing.
To walk into any public space and just look at the folks that are there, not through a judgmental lens, but just asking yourself, who here is doing hard things? And what's interesting is you'll see body compositions and body structuring and people going downstairs and people trying to go upstairs. And all of a sudden, you start to see the world through this lens of people that are doing hard things are moving effortlessly up the stairs. They're gliding down the stairs. People that aren't are hunched over. They're hanging onto the rail. They're doing these other things. And they may be in their 40s. They're not even like elderly people, right? And so this idea of doing physically, not only, intellectually or business-wise or relationship-wise or anything you want to apply it to, but doing things that are physically demanding is absolutely essential for health in every level, whether it's bone density, muscle mass, brain health, all of it, right? But I think it's interesting because people have talked about exercise being good for the brain, but nobody really knew exactly why. Well, you improve blood vessel flow and you improve oxygenation and you keep your arteries healthier. But this whole BDNF story is kind of interesting too. So if you're in the audience, think about doing some things that are challenging to you. Cultivate a love for that. There's a big trade-off.
Absolutely. And it's not just your cognition. The BDNF pathway also affects depression, which makes sense because of brain inflammation. And it affects so much of your health. And I had mentioned that about 4% of the population have 2 copies, but about 30%, so a third of you out there have 1 copy. And 1 copy alone, having of that grouping, can still give you 2.7 times the risk of cognitive decline. So that's still a 170% increase that you can fix with movement. And most of those people, once they start moving, they feel better. They really notice a change.
Exactly. Oh, that's really, really cool.
This episode of the Gladden Longevity Podcast is brought to you by Mimeo, a supplement designed to give people the benefits of fasting without having to fast.
Yeah, Mimeo's research found that the human body produces a unique set of molecules during a 36-hour fast. And those molecules were responsible for activating many of the cellular health and longevity benefits of fasting.
With Mimeo, you can get the cellular health and metabolic benefits of a 36-hour fast even during a meal, or you can use it to maximize the benefits of your shorter fasts with powerful boosts in mood, focus, energy, performance, recovery, and appetite control. Dr. Gladden, in the 2 years I've been working with you and the 5+ years I've been listening to this show, I've never heard you talk about something outside the Gladden Longevity line of products like I've heard you talk about Mimeo. Why do you like it so much? Use it and make it a part of some of your presentations.
Yeah, we love it, quite honestly. I mean, the idea of Mimeo being able to mimic fasting, we all know the benefits of fasting, is dramatic in and of itself. And even in the context of if you're eating, and if you're fasting, it even augments your fast that much more. But the interesting thing is that it also decreases things like neuroinflammation, it suppresses appetite, and it increases a very, very important biological process called reverse cholesterol transport. And that's critical for cardiovascular disease, for decreasing risk of dementia, cancer, et cetera. It's really, it's quite a fascinating product. I've really been kind of blown away by it.
As a way to help support this podcast and save money on your Mimeo order, you can use the discount code Gladden10 to get 10% off your order at mimeohealth.com. That's that code again, mimeohealth.com. That code again, Gladden10 at MIMIO health.com.
Think of it as a way to unlock the power of your biology. So we've talked a little bit about brains. Do you want to talk a little bit about, we've talked a little bit about diabetes and metabolic disorders. Do you want to talk a little bit about cardiovascular stuff?
Yeah, absolutely. And we can also talk about, since you are the longevity doc, if you want, we can also talk about some of the genes that relate to longevity. But there are some that, many of the genes that relate to cardiovascular health also relate to longevity, which makes sense. So,
you know, I'll just interject this. In the work that we're doing, you know, we're doing this transcriptomics and proteomics that I mentioned to you the last time we were together, where we're looking at the genes and then we're actually doing the proteomics and transcriptomics to see how the genes are actually being expressed. So it's one thing to kind of know what the genes are, but then how are they actually being expressed, right? So there are downstream effects that you can look at in terms of, well, your insulin resistance or cognitive decline or whatever, but it's nice to kind of look to see, are you impacting the way these genes are really being expressed? So in doing that, one of the things that we found that seems to be a central category in the aging process is excess inflammation and oxidative stress. And so when we attack that, it seems to help everybody, right? When we're really attacking inflammation and oxidative stress, whether it's brain health, whether it's heart health, almost anything. And so, you know, inflammation has many sources. You know, inactivity is one, but also as we age, senescent cells form and they release inflammatory cytokines. The immune system, as it ages and adds glycan molecules to the ends of the IgGs, can increase the inflammatory predisposition of the immune system. So now all of a sudden you have lots of things kind of ganging up into this whole inflammatory oxidative stress pathway that really starts to tear things apart. And just so the audience understands, the real problem with inflammation is that it increases oxidative stress at the cellular level, which then starts to destroy components of the cell, either damage the DNA, the proteins, or whatever else. So anyway, what we're talking about here is so central to almost anything that you want to try to optimize. You have to know your genetics here, and you have to be able to go after it and then sort of see how you're progressing. So pretty interesting.
Yeah, well, I think that those are really good topics of inflammation and oxidative stress and the intersection with longevity and cardiac disease, because two of the most common, well, not most common, two of the best known and best studied genes that have the biggest impact on longevity are FOXO3 and Klotho. And FOXO3, it's a nutrient sensor that's associated, it's actually a benefit. If you have 2 copies of FOXO3, you're going to have lower risk for heart disease and lower risk for mortality. So that's why it's a longevity gene. But it works in part because it decreases inflammation. We had talked about IL-6. Well, IL-6 and TNF-alpha in particular are decreased when you have higher FOXO3. And those are just big names for, you know, different inflammatory pathways. But again, those are pathways that ultimately can lead to tissue inflammation, which can cause tissue destruction essentially, and even scarring. So they can lead to something, again, lots of names, called FOXO3. When you don't have enough of it, you get higher TGF beta and you can have more scarring of tissue. So by lowering the inflammation and lowering scarring, you live longer. Oxidative stress is another topic. Klotho is a protein that helps us protect against oxidative stress. And people who have 2 copies of that gene, it's not good. They don't make enough Klotho, and they'll age prematurely, almost like as if you leave something out in the sun. That's the way to I describe oxidative stress to my patients, is if you leave something out in the sun, it's going to deteriorate faster because the sun is a big source of oxidative stress. So furniture left outside in the elements is going to not look the same after 10 years compared to something in your house. And Klotho is that ability to repair that stress. And you go, well, how do we get oxidative stress? Well, by making energy. So if you think about, if we have a nuclear power plant, we always have to go and deal with the nuclear waste. And when we're trying to make energy from oxygen, we make this oxidative stress that, as you mentioned, if you don't have enough of it, it'll blow up your mitochondria. And one of the things is people who have low Klotho, they should do aerobic exercise before doing resistance exercise. Because if you do so I know that a lot of people will alternate and they'll do resistance one day and aerobics another day. But if you're in that 3% of the population that have the ultra low Klotho levels, they've shown that resistance exercise will further lower your Klotho. So it'll further accelerate your aging, whereas aerobic exercise increases Klotho. And then there's other things that can increase. There's various supplements. Omega-3s help a little bit, but resveratrol can help with Klotho. And then there's some other things like from chlorophyll and from hops, but you can't do it the beer way. The alcohol kind of negates that effect. But there's lots of different things that, and knowing it can help. And you can't with aging just go, well, my parents lived a long time, so I'm going to live a long time, because some of the genes having 1 copy is a benefit. And some of the genes having 2 copies is a risk. It's not quite simple math with genes.
That's right. Yeah, and the Klotho story is pretty interesting. Also, I heard a lecture some years ago where someone from UCLA, and I'm blocking on the name right now, but I'm not sure I ever really knew the name. But the point is that they were talking about if you were heterozygous for Klotho, that you actually stood in the best stead, if you will. So being homozygous for either low Klotho production or for high Klotho production, being in that middle point somehow was the best spot. Do you have any thoughts about that?
I do. So we didn't have Klotho in our report until this year. And it's because we won't put something in the report till I've figured it out enough to be able to explain it and teach it. And that was what I was going, this doesn't make sense, because having 1 copy, is a benefit, but 2 copies is not a benefit, even if it's, and so what it turns out is there are 2 different variants. So there's 2 different SNPs in Klotho, and one makes you have more and one makes you have less. But when you have 2 copies of the one that makes you have more, it is more than 99% of the time co-inherited with 2 copies of the one that makes you have less. And the less wins, and the less is really bad. So that when you have 1 copy, the way it works is that gene affects the signaling and the promoter, and you don't have the turning off. It'll help you to make more because it affects the transcription. So that is the highest transcription. That's the highest Klotho levels, is when you have 1 copy of each. And it's because those 2 genes, the one that makes more and the one that makes less, are co-inherited. And that's the kind of stuff so that's where, I always say.
Are you reporting on 2 Klotho genes then?
Mm-hmm. And so there's a benefit and a risk. And the real thing is, the benefit is having 1 copy of each, and the risk is having 2 copies of the one that makes you have less Klotho.
Right. Okay. Yeah, that's fascinating. Yeah, the interesting thing is that it's the minority of people, as I recall from that lecture, that actually have the heterozygous configuration. Do you want to speak to that? Did I get that right? I'm not sure.
Correct. So, and that means that most people have an opportunity to improve their clotho status. Because if you look at, we can't see here, but if you look at pictures of rats that have low clotho, a rat that is young will look twice its age and they'll die earlier. And in fact, the people who have the lowest clotho have a lot higher chance of not living beyond 75. So it's about 25% of the population that get that beneficial one of each. So for the rest of us, only about 3% get the really low, but then the rest of us, the other 60%, they're kind of in that medium, they have low, but not super low. But that means that for most people, things that improve clotho, the aerobic exercise, resveratrol, omega-3s, all of the things that help other oxidative stress pathways, like algae is a great source. Some people like little algae bits and such. Those are going to be benefits for them.
Yeah. Spirulina type thing.
Spirulina, yeah, it's really, it's the reason when you use things like spirulina for oxidative stress, it's not specific for clotho, but clotho regulates all these other oxidative stress pathways. And so you're going to have lower of all the antioxidants. So you can use basically any of those antioxidants to kind of respond to low clotho. Or the most ideal, the most direct, is to increase clotho transcription. And like I said, the most specific for that was resveratrol. And then there are some things like xanthahumol and omega-3s that also help somewhat.
Yeah. So in our work too, we're actually starting to work with custom-built peptides to go in and modulate gene expression. So this may become a target for us in our next round. This LifeRaft index that we've developed, this testing index, is to measure clotho for people at the productivity and then be able to go in and modulate it. And the interesting thing is that when you do that, you actually change the expression, not only in the cells that the peptides are affecting, but in the daughter cells, which is really interesting.
That's great. And I think that one of the great things is, one of the limits, of course, of proteomics and metabolomics is not everything's expressed in the periphery, but clotho is. So there are some things that are harder to measure. And that's why I think the combination of proteomics, genomics, all these different things can be beneficial, because inflammation, for example, could be more specific to tissue.
Do you want to switch gears for a second and talk a little bit about bone density? Because we see a lot of people, regardless of their age, that come in to see us, and we do DEXA scans for virtually everyone also, unless they have some contraindication to it. And we see that a lot of people that really aren't that old, maybe in their late 40s or mid 50s, or have significant bone loss, which is pretty dramatic. My bone density has always been excellent. And in fact, I just had it tested a few months ago, and it was in an above average range for somebody in their late 20s, right? I attributed that to the fact that I wore lead in the cath lab for, 25 years. But I haven't worn lead in, a dozen years, and the bone density is still good. But I do exercise frequently. So do you want to speak a little bit about that?
Yeah. And so definitely, that idea that you can have good genes, there definitely are genes that affect bone formation. I was speaking with a doctor who is a very well-known integrative functional doctor, has taken great care of himself. And he was talking about, kind of for fun, when he was in his 50s, well, not really for fun, but they did a bone density trying to be a complete workup of his body. And it was low when he was in his 50s, even though he had been young and active. And when we got back his genes, he was in the 1% of the population that had this gene that related to bone formation, though he never made enough osteoblasts. So there are some people who are genetically predisposed. But again, the really cool thing is if you learn that relatively young. And so genomics isn't just for people that are having cognitive decline or seeing their bodies start to fail. The best time to do genomics is learn about your genomics in your 20s or 30s or 40s. We even have babies learning about their genomics while their parents are learning, because you can use alpha lipoic acid and ashwagandha to kind of further stimulate those osteoblasts, which are bone building cells, if you have that gene. I think the biggest thing about genomics is there's different causes for different people. So I have a strong family history of osteoporosis and I'm a runner. I'm not a very fast runner, but I'm still a runner. And my bone density is not great for a woman in her 50s, but I'm in the 1.7% of the population that has a different kind of collagen. And so what I do for my bones can be different than what the physician did for his bones, because we have different contributing factors. And for me, it's also about over removing old bone. So I think that that's the take home point for genetics, is that for any topic, for heart disease, there you can do baby aspirin, you can do nattokinase, you can do curcumin, you can do all these on and on and on, statins, all these different things, both prescription and not. And for every topic, that's the case. You can do sulforaphane for some genes for the bones. You can do collagen. You can, and I think that, so the way that I think about it is when we build bones, our bone density is not just what matters, it's also our bone quality. So how well are the bones hooked together? And that's a lot based on estrogen levels. And even men have to have estrogen. So even your genomics of how well you convert testosterone into estrogen affects it. But of course, also lifestyle, because if you have too much iodine, you won't convert to estrogen. If you have too little iodine, that can affect other things. If you have, again, too much inflammation, that can affect your bones as well. So I think the bottom line is we are complex. Of course, we would expect humans to be complex machines. And so if you can understand, the more that you can know about what are those things that you're in the 1%, the 2%, the 5% of, can really help you to tighten up what I call your own personal protocol for both diet, lifestyle, and food. I mean, we've talked about exercise and diet, exercise and supplements, but there's also really specific foods for different genes that can help turn on and off genes.
Yeah, no, exactly. I think, when you think back to the traditional medicine that you practice and you look at the double-blind placebo-controlled trials of anything across 10,000 people, and they never know the genetic profile of those people. They only know their ages, let's say, and their sex, so to speak, and maybe a little bit about their cultural context, right? But they don't really know who they're testing. And then they're trying to decide, well, this is a good thing for heart disease or diabetes or whatever else based on these results. And it's so random in so many senses that, just so the audience understands, to really get this right, you have to think of yourself as really, I think, as a complex ecosystem, kind of like a rainforest. And it's really not about trying to, I'm an aging man, my testosterone is getting low, so I'm going to take testosterone. And on top of that, I'm going to take an anastrozole because I don't want to convert to estrogen. I don't want to be a woman. I want as much testosterone as I can get. Well, in that thought process, you're taking that ecosystem and now pushing it out of balance. And what happens is, you don't have estrogen. So now you're compromising your brain. Now you're compromising your bones. Now you're compromising your belly fat and things like this. So it's really so important as you're thinking about this. If you really want to optimize your health for the long term, you need to be working with somebody that's looking at all the factors and putting the ecosystem back into balance. That's really what you're going for. That's the way I think about it. So
I absolutely agree. I think that people tend to look for their own answers, and I'm all about becoming self-aware, but we are complex. And I do think that a trained physician can really help because, as you said, like a lot of people will try to take anastrozole or way overdo it on the iodine because they want to keep their testosterone high and their estrogen low. Well, they've shown if you block a man's conversion of testosterone to estrogen, again, you'll get more brain inflammation, you'll get more bone loss because it's not testosterone that protects the bones, it's the estrogen. And you don't want to have women's levels of estrogen. You don't want to have an estrogen level of 100, but you do want one that's over 20. You don't want an estrogen level that's undetectable as a man. There's so many other fun things that can be learned from genomics. There are people who want to do everything naturally, and then there are people who want to do things with medications. And it doesn't matter which way you want to do things, genomics can help. There are people who really need, you're a cardiologist by training. There are people who really need statins. They have high levels of cellular adhesion molecules and things that make plaque that are known to be associated with heart disease. And maybe they can't do it with diet. There's genetics involved. Well, if you're somebody who doesn't make enough CoQ10, if you're somebody who doesn't metabolize statins right, all of that can be helpful because it can allow you to change your dosing. And one of my friends who went back to school and was becoming a healthcare provider, was saying, oh no, this is the kind of the way that we're taught, that this is how we do hypertension. This is how we do diabetes with these algorithms. And generally the algorithms work if you happen to have the most common genes as your cause. But treatment-resistant hypertension, it's nothing. It can be treated with one drug. It's just about knowing which is the right drug. It's usually that it's somebody who makes high aldosterone because of a gene and they need this other medicine.
We just went through this in my practice where somebody came to us on 3 or 4 medications and we ran their genetics and, oh, there it was. This guy needs spironolactone because he's a big producer of aldosterone, which, just so the audience understands, is something that's called a mineralocorticoid, which basically causes your kidneys to retain sodium, retain fluid, and tend to drive your pressure up. And you can put yourself on lots of blood pressure medications, but if you don't know you have that and you're not on the right medication, which actually counteracts it directly, you can spin your wheels for years and bear the consequences of it. So this idea of being precise for the individual is everything, I think.
I agree. And it's so much fun because there's a measure, and again, it's called proBNP, of how well the heart squeezes. And I've had patients come to me almost in full heart failure. That number should be less than 100. And in someone young and healthy, you'll see it, 30, 50, even if they're older, they could be 70 years old, but they could still have a great heart squeeze. And I've had patients that come to me at 700, and with their genomics, we're able to see what's going on and guide them and get that down to less than 200 and sometimes less than 100. And so we get huge drops, and that's going to really impact quality of life, because when your heart isn't squeezing, you're going to feel when it doesn't squeeze well, you're going to feel short of breath with exercise. You're not going to get your whole bang for the buck. So for me, I think that medicine was getting frustrating 15 years ago, even 10 years ago. And I was looking for ways to make it not only more enjoyable, but more efficacious. And I wasn't finding them. Even having longer appointments, it wasn't giving me that satisfaction as a doctor until I delved into the genomics in this way and developed IntellexDNA and was able to improve my outcomes. And that gave me satisfaction, because we all go to medical school wanting to help people. And, that's like the classic med school essay, oh, I want to help people. And then we get there and go, okay, we're not helping people stay healthy. We're being, technicians to fix them once they're sick. And how do we really help stay healthy? And that's where the game changer has been, is being able to untangle people with chronic pain or brain fog or who just don't feel well. Now I have a tool. And that's what's been so exciting for me.
No, it is exciting. I mean, I tell people all the time that our greatest joy is seeing our clients do well. And there's no greater joy than that, right? So, and having tools like yours to help us do that has really been a game changer. So I can't thank you enough for developing this, and the fact that it's under ongoing development, that you keep adding panels, looking at adding more genes like you have this year and things like that. So it's really been such a pleasure. So thank you for all that.
Thank you for being there since the beginning, Jeff. You've been using this now you were one of our early adopters, because you are one of those doctors that's at the forefront of trying to use the best things out there. But with you as a pioneer, it has been great having you help lead the way.
Oh, well, I appreciate that. We appreciate it. It's been super fun.
Thank you for listening to this week's episode of the Gladden Longevity Podcast. If you would like more information on what we've discussed or other topics, please reference the show notes or go to GladdenLongevityPodcast.com. You can also find us on Instagram, Facebook, and Twitter by searching Gladden Longevity. If you've enjoyed this podcast, please subscribe to get future episodes delivered to you, and share our podcast or this episode with someone in your life that you may find benefit. Thank you for listening. We'll be back next week with another exciting episode. The Gladden Longevity Podcast is provided for informational purposes only. It does not constitute medical advice. This content is not intended to be a substitute for professional medical advice, diagnosis, or treatment. Always seek the advice of a physician or other qualified health provider with any questions you may have regarding a medical condition. The use of any information and materials linked to this podcast is at the listener's own risk.